Hello, my name is Merle Fuchs. I'm the CEO of PRAMOMOLECULAR. PRAMOMOLECULAR is a spin-off of the Technische Universität Berlin, founded by 1.7 million euro via Access Forschungstransfer and business angels. We have developed a platform technology that enables for the first time to silence genes efficiently in the lung and in the pancreas. And therefore, we are developing drug candidates against pancreatic and lung cancer. Our team of 3 consists of technologically and entrepreneurial, very experienced founders. In large parts, we are a virtual company, cooperating with experts who are among the best in their fields. This gives us the possibility to be very fast, to be flexible, to have high professionality and high development speed. We are working with siRNAs. That's very important because most of the disease-causing proteins cannot be targeted by classical drugs, so-called small molecules, because of steric reasons. But siRNAs, small interfering RNAs, can theoretically downregulate every disease-causing mRNA, even of proteins that cannot be targeted by classical small molecule drugs. An siRNA, if an siRNA binds to their covalent mRNA, it catalyzes the downregulation of the mRNA, and therefore the protein the mRNA is coding for cannot be formed anymore. By this mechanism, theoretically, every disease-causing protein can be downregulated if the siRNA can reach their target cells. Small interfering RNAs are safe and they are efficient, but they are unstable. And therefore, they have to be combined with delivery systems for stabilization and for transportation. Normally, RNA-based active ingredients like RNA vaccines or siRNAs are packed in nanoparticles. But the formulation of these nanoparticles is a very complicated process and the limiting step, for example, in the RNA vaccine production. We don't need this particle formulation. We just combine by covalent chemical bonds 2 to 3 lipids to one end of this double-stranded siRNA. These lipids work as transport systems and they work for stabilization. By this mechanism, we have the simplest in vivo delivery system for siRNAs known to us worldwide. These self-delivering siRNAs are stable and they are effective, and they can be stored if lyophilized at room temperature. But our most important USP is, for the first time, we are able to in vivo silence genes in the lung with our delivery system PRAMO-01 and in the pancreas with our delivery system PRAMO-02. This is very important because up to now, gene silencing only worked well in the liver or in local applications, for example, in the eye. The next slide, please. Can you help me, please? We have the easiest technology for the rational design of fully automated synthesis of gene silencing drugs for lung and pancreas. This enables us to build up a broad pipeline of drugs against diseases with high medical need. And our future potential is our technology enables the fully automated GMP-compliant synthesis for precision oncology at the point of care with drug printers. Next slide, please. We're addressing oncology first. Oncology is the largest pharmaceutical therapeutics market. And still, oncology is one of the most important drivers for mortality. And lung and pancreatic cancers are among the deadliest cancers worldwide. Next slide, please. We are addressing RAS oncoproteins. They are the most important family of oncoproteins. And our first drug candidates are KRAS mutations. KRAS mutations are the oncogenic drivers of 90% of all cancer diseases, 32% of lung adenocarcinoma, and 97% of pancreatic cancer. Next slide, please. In our in vivo proof of principle, we could show efficient delivery and silencing in the lung and in the pancreas. We were able to downregulate housekeeping enzymes in the lung with 50% and with 40% in the pancreas. These experiments were done with unmodified molecules. Next slide, please. In our proof of concept experiments, we work with stabilized and optimized siRNAs. And in vitro, we could show in human lung cancer cells a downregulation of 80%, as you can see on the left, and on the mRNA level, and on the right, you can see that we are also able to downregulate the corresponding protein. Next slide, please. In our in vivo proof of concept experiments that are currently running, we try to downregulate the KRAS wild type, but also human cancer cell lines in different xenograft models, and we are measuring tolerability, efficiency, and biodistribution. Next slide. We have filed our patent for the delivery molecules at the end of last year. A good research answer and the FTO analysis indicate high probability of grant. We will protect all promising drug candidates via international additional patent applications. Next slide, please. Our business model, together with pharmaceutical partners, we will develop our drug candidates up to clinical phase 2B. And for orphan applications, we aim to develop our drug candidates till clinical phase 3 by ourselves. An exit option would be in trade sale, but we would prefer an IPO. Our most important competitor is the DTX Pharma from California. DTX is also developing self-delivering siRNAs, but their technology is much more complicated than ours. DTX aims at applications in the eye, in the neuromuscular and in the muscular system and in the central nervous system. We aim at diseases that have a much higher market potential than that of DTX. DTX had been founded in 2017, and they are financed with $150 million yet. But we think they are technologically at least 1 year ahead of us. Because siRNAs are safe and able, we really hope and we see a good chance to reach our target product profile. If we are successful, our licensee has the chance to reach a market potential, a sales peak of 750,000 euro in 2032 with a market share of 26%. This year, this year, we want to show our proof of concept in the lung, next year in the pancreas. And to reach the first in man in 2032, to do this, we need 55 million euro, and we hope to close a Series A finance in the beginning of next year. Thank you for your attention. Any questions?
Yeah.